Hello and welcome everyone to this video podcast for Evolution and Diversity. We just finished talking about bacteria and archaea. Now we're going to move on to the next domain of life, that is the eukaryotes. In this video, we're going to start by talking about eukaryotes as a domain of life and providing some basic introductory information about eukaryotes. And then we'll talk about endosymbiosis as an explanation as to why eukaryotes have mitochondria and chloroplasts. Then we'll talk about evidence for this endosymbiotic theory that explains why mitochondria and chloroplasts are found in eukaryotes. And then we're going to close with talking about the evolution of the nucleus. I'd like to begin with this brief timeline of the history of our Earth. As a reminder, Earth is about 4.576 billion years old. At the beginning of the semester, we talked about chemical evolution and how that led to the formation of chemicals that would be necessary to sustain life. Following the chemical evolution, many other events occurred, but ultimately it led to the formation of life. And we have evidence that life first formed on Earth about 3.5 billion years ago, because that's when we see our earliest fossils. And at the close of our last unit of material, we talked a little bit about how some bacteria, the cyanobacteria, were able to use light as energy and to produce their own carbohydrates. And as a result, they produced a lot of oxygen in the atmosphere. As you recall, when this happened, several things occurred. Many species died out because of so much oxygen in the environment, which then led to an ice age, which then, after the ice age, led to an explosion of new species forming. And that's where we're going to pick up the story here, how eukaryotes arose, and how we define what a eukaryote is based on what's inside of those cells. And eukaryotes have existed ever since. And we can see other landmarks, like the first hard-shelled animals, and we can see our friends the dinosaurs that we're familiar with. And as also a reminder, right, humans are at this very end. Humans have occupied this Earth for a very minimal amount of time compared to the age of the Earth and compared to when life first formed on Earth. And we're going to spend the rest of the semester talking about this time period here. But first, it's important to think about what is a eukaryote, and then how did some of those early parts of the eukaryotic cell, the organelles, initially form. So let's begin with this phylogenetic tree that we've become familiar with, where we have our bacteria coming off this first node, and then we have our archaea splitting off here, and then the eukaryotes. We spent a lot of time already talking about bacteria and archaea, and now we're going to start talking about what happened here to allow eukaryotic cells to evolve. So some key features of eukaryotes, and certainly this is not an exhaustive list, but they tend to be larger than bacteria and archaea cells. While we talked about some primitive organelles in bacteria and archaea, eukaryotes all have organelles. Now remember, an organelle is a membrane-bound structure within the cytoplasm of the cell. And each organelle has a very specific function. It allows the cell to compartmentalize and specialize in certain functions in certain parts of the cell. Eukaryotic cells also all have a more specialized cytoskeleton. We talked about how bacteria and archaea have a primitive cytoskeleton, but when we think about eukaryotes, they have a much more elaborate and much more specialized cytoskeleton. And remember, the cytoskeleton are these proteins that organize within a eukaryotic cell that helps in moving material within the cell. It helps the cell move in some cases. It helps make the overall form and structure of that cell. It has lots of different functions that you'll learn more about when you take cell biology. They have a nuclear envelope. So all eukaryotic cells have a nuclear envelope, which makes up the nucleus. With eukaryotes, we see multicellularity. Now, not all eukaryotes are multicellular, but many of them are. And the last thing I'm going to say here has to do with cell reproduction. Now remember, in bacteria and archaea, cell reproduction occurred by binary fission. In eukaryotes, cell reproduction occurs in two ways. One is through mitosis, which is a form of asexual reproduction, and the other is through meiosis. And in meiosis, gametes like sperm or egg, or in fungi, spores are produced. And these are used in sexual reproduction. I'm going to spend a little bit of time now talking about some of the organelles. So here what you see is a fairly standard picture of an animal cell and a plant cell. For this class, you do not need to memorize all of the different structures of a cell. And I'm not even going to go over all of the different structures or all of the different organelles. But we are going to talk about some of them throughout the rest of this semester. And when we do, then we'll define them and give a brief description of them. Today, we're going to go over how the nucleus was formed, how the mitochondria was formed, and how the chloroplast was formed. So I think it makes good sense to talk a little bit about each of these, at least as far as what their functions are. And again, when you take cell biology, you'll go into much greater detail. For now, though, the nucleus you should know as the site where chromosomes are stored. In fact, to make this a little bit more specific and correct, we should say where most chromosomes are stored. You're going to learn that there are other sources of DNA, chromosomes in the cell, and they can be found in the mitochondrion and the chloroplast. The mitochondrion. The mitochondrion, as you may recall from a previous class, is where most of the chemical energy is made. And by chemical energy, I mean ATP. The mitochondrion also plays an important role in apoptosis. Apoptosis is a cellular event that you'll learn about later that is essentially cell death. It's a program within the cell that triggers it to die. And we do want our cells to turn over. We don't want our cells to live forever. It is also important in storing calcium and in steroid production. Next, let's talk about the chloroplast. Its biggest claim to fame is that it is the site of photosynthesis. Now, you won't need to worry about the details of photosynthesis in this class, but you should know the general idea that you take CO2 plus water, and with the source of light from the sun, oxygen and carbohydrates are made. You should also know that the chloroplast is important in providing immunity to the plant. Plants have to do a lot of very cool things because they don't have some of the features that an animal has. Right? They can't move. They don't have an immune system. And so the chloroplast helps provide some of this innate immunity for a plant. And we'll talk more about that later in the semester. It is also the site of amino acid synthesis. Almost all of the amino acids in a plant are made in the chloroplast. I'm going to talk a little bit about multicellularity. It's important to remember that going from a unicellular organism to a multicellular organism is one of the most impactful evolutionary adaptations that we've seen. It's important to remember that it did not go from having a single cell here to having a fully formed multicellular organism like humans in one step. And that's why I wrote up here many different arrows indicating that it took many different steps. And in many cases, we don't really know what each of these steps were. We can make some speculations, we can make some educated hypotheses as to how some of these events occurred. But what we know is that it did take multiple steps here. Essentially, what we think happened was that you would have had a few cells that were unicellular, and they might divide, right? And usually when a unicellular cell divides, you produce two cells. And they go off and produce two more cells. But they are separate from each other. What we think might have happened here is that when one of these cells divided, instead of separating from each other, they stayed as a single unit. Still two separate cells, but as a single unit. And then as they divided more, you would increase this clump of cells until you had a larger clump of cells. This larger clump of cells now could operate together. And then at this point, selective pressure could have been placed on it. Maybe some of these clumps of cells were able to outcompete other clumps of cells, which I'll just put up here as a different clump of cell. We don't know why it was able to. Maybe it was able to reproduce faster. Maybe it was able to find food faster. Whatever happened, one of these clumps of cells was more fit than the other clump of cells, and so it survived. As time went on, these cells might gain some specialization. which I'm just going to show as different colors. The reason this was more competitive than a competing multicellular structure here, as I said, was because maybe some of these became specialized. And so maybe we had some cells that were specialized to find food. Maybe some of the cells were specialized in movement. Other cells might have specialized in communication. And yet others specialized in reproduction. And we could make a longer list as well. But the idea, when you take away from this, is that you had this clump of cells that stuck together, which was much different than what we saw before. And then over time, selective pressures allowed some of these cells to specialize. And by specializing, the organism was able to compartmentalize certain functions to certain types of cells. Every cell didn't have to do the exact same thing now. Now, this ability to specialize like this is centered around gene regulation. The ability of each of these specialized cells to turn on and turn off a specific subset of genes was critical. Because remember, each one of these cells has the exact same DNA in it. Just like you and me, every one of our cells has the exact same DNA in it. But only certain genes are on. So like for you and I, our liver cells need to turn on liver genes. And we don't want our liver cells to turn on brain genes. We want our brain cells to turn on brain genes, but not liver genes. This is critical in the formation of this specialized multicellular organism. And ultimately, this specialized set of cells in this multicellular organism here would ultimately evolve into the many different specialized species that we see today within eukaryotes. Now, while this set of events seems almost remarkable that this could occur, evolutionary data suggests that it's occurred multiple times. So multicellularity evolved one time in animals, 3 independent times in fungi, and then 6 independent times in algae, all with a similar result in having a multicellular organism with specialized cells. As we begin this discussion of eukaryotes, I'd like to focus on one group of eukaryotes that we call the protists. And protist translates to the very first. These represent some of the very first eukaryotes that we know of. And as you can see in this phylogenetic tree, one of the first characteristics of a protist is that they are hard to classify. And that is because we see our 3 kingdoms of eukaryotes, fungi, animals, and plants, and then we see intermixed here many of these other lineages of eukaryotes that are not fungi, animals, or plants. They are different. They are the protists. But as you can see when you're looking at this, they do not form a monophyletic group. And so the best way to characterize a protist is to say that they are any eukaryote that is not an animal, plant, or fungi. Since they are not found in a single monophyletic group, there's not a single trait that unifies all protists. When we investigate plants, animals, and fungi, we'll learn that all of these have specific traits that are only found in that specific lineage or in these specific kingdoms. Protists, not the case. The only thing that comes close to linking them all together is that they're always found surrounded by water. Either wet soil or in an aquatic habitat. However, that trait is not only found in protists. We see many examples of fungi, algae. We see many examples in fungi, animals, and plants where they are near and surrounded by water. Near or surrounded by water. Okay, I'd like to move on to our next objective, and that is to talk about endosymbiosis. All right, let's go ahead and move on to our next objective, where we're going to talk about endosymbiosis and how this has led to a theory to explain how mitochondria and chloroplasts evolved. So first, I'd like us to consider this bacterium. And this bacterium would have its DNA. It would also have several ribosomes. Along the membrane here, the plasma membrane, was the electron transport chain. And then on the other hand, we have a eukaryotic cell. And this eukaryotic cell has its chromosomal DNA in its nucleus. It has several organelles like mitochondria. And if it were a plant cell, it would also have its chloroplast, as well as other organelles. It would also, of course, have ribosomes in the cytoplasm. So how did we get from a bacterial cell to this complex eukaryotic cell? As often when we talk about evolutionary events, it's important to remember it wasn't this one single event that happened, right? It was several events that eventually led to the establishment of this complex cell. What I want to do over the next several whiteboards here is talk about first how the mitochondria evolved, then how the chloroplast evolved, and then finally how the nucleus evolved. As we get started thinking about the evolution of these organelles, let's break down this word endosymbiosis. First, let's think about the word symbiosis. This is the close and prolonged interaction between two different species. Now, symbiosis could be beneficial between the two different species. It could also be beneficial for just one of the two species. It could also be harmful to one of the two species and beneficial to the other. And when you take ecology, you'll break down these different kinds of symbiotic relationships in more detail. But for now, I want you to think about symbiosis as just this interaction between two different species. Next, let's think about the term endosymbiosis. This is a type of symbiosis, but it's a very specialized type. This occurs when one organism lives inside a cell of a different organism of a different species. Now, thinking about this idea of endosymbiosis, scientists were able to establish a hypothesis to explain how mitochondria formed. And that hypothesis later became a theory. And that theory is called the Endosymbiotic Theory. Now, scientists in the early 1900s started thinking about this idea, but it wasn't until 1967 when Lynn Margulis was able to advance this hypothesis and get a lot of other scientists thinking about it. And ultimately, enough data was gathered over the next decade or so to bring support to this idea of endosymbiosis as being the theory to explain the origin or the evolution of mitochondria. So let's put some words to this endosymbiotic theory to explain what it is. So approximately 2 billion years ago, an aerobic bacterium was taken up by another cell. And then this aerobic bacterium became the mitochondrion. Remember, an aerobic bacterium is one that can utilize oxygen through the electron transport chain to generate chemical energy. Now, what kind of cell took up this aerobic bacterium? Well, that's somewhat debated. So this other cell here may have been an amoeba-like eukaryote, where here's your amoeba cell, and then here is your bacterial cell. Amoeba, to survive, they take up other cells and other debris in order to digest them and survive. So it's not unreasonable to think that bacteria would have been engulfed by this amoeba-like cell, like so, where this bacteria that will eventually become a mitochondria contains the bacteria surrounded by a second membrane, that of the amoeba, the plasma membrane of the amoeba. This way, this amoeba-like cell, which already likely had a nucleus, was able to outcompete any other amoeba-like cell that didn't have the ability to use the oxygen. Because we know that cells that can use oxygen through the electron transport chain and through cellular respiration are going to be able to generate a lot more chemical energy in the form of ATP. One could imagine how that might make this particular cell evolutionarily more fit than a cell that could not undergo cellular respiration using the electron transport chain. Now, we don't know for sure if this is exactly what happened, but there is evidence to support that this is a likely explanation as to how a mitochondria could have formed. Now, another idea is that maybe there was a bacterial cell that engulfed another bacterial cell, or maybe even an archaeal cell. And then this bacterial cell had the archaeal cell inside of it. And this archaeal cell would then become the mitochondria. I should also draw this showing that the mitochondria would have two membranes as well. Whatever the case may be, the same general principle applies, that one cell took up an aerobic bacterium that then would become the mitochondria. And as I said, when this idea first came out, it was just a hypothesis. And for an idea to move from being a hypothesis to being a theory, one needs to gather evidence from multiple fields that support that hypothesis. And over time, it becomes widely accepted in the scientific community and becomes a theory. And that's what happened here. So what I'd like to do now is talk a little bit about the evidence that supports the endosymbiotic theory for the explanation of mitochondrial evolution. One of the more compelling pieces of evidence is that today we see alpha Proteobacteria in an endosymbiotic relationship with Protists. In fact, there are three major groups of Proteobacter that only exist inside of a protist. And they have to exist inside of the protist in order for the protist to survive. So we see direct evidence that bacteria can live in a eukaryotic cell, a protist. Some other evidence is that mitochondria and alpha Proteobacteria are about the same size. There are some differences, but roughly speaking, they're about the same size. Mitochondria and bacteria both divide by binary fission. They do so in a way that's independent of the division that is happening within the nucleus, that is independent of mitosis and meiosis. Mitochondria have ribosomes, and those ribosomes make proteins inside of the mitochondria. In fact, the ribosomes found in the mitochondria are more similar to bacterial ribosomes than they are to eukaryotic ribosomes. Mitochondria have their own genomes. Like bacterial genomes, mitochondrial genomes are circular. Mitochondrial genomes code for proteins necessary for electron transport. Mitochondria have double membranes. Now, bacteria don't have double membranes, but if you remember what we said about that endosymbiotic event, where this eukaryotic cell engulfed that bacterium, like so, and then when it brought it inside of the cell, it encircled it with its own membrane. So you would have ended up with a cell that looked like this, with the bacterial membrane on the inside. So the inner membrane, which I'm just going to call IM, is bacteria. And then this outer membrane, OM for outer membrane, is eukaryotic. So the fact that mitochondria have two membranes suggests that such an event could have occurred. What gives this more support is remember that this bacteria, before it was engulfed, and all aerobic bacteria, their electron transport chain is on their plasma membrane. So one would predict, if that's true, that this inner membrane of what would become the mitochondria would be the site of electron transport. And it is. So these 6 pieces of evidence provided a lot of support for this endosymbiotic hypothesis. However, there was one competing hypothesis that still could not yet be disproven, and that was called the within eukaryote hypothesis. And what this hypothesis argued was that you had a eukaryotic cell with its nucleus, and that a double membraned structure formed here that, like the other idea, would also have formed from the plasma membrane of the cell. But the problem with this idea is there's no DNA. And we know mitochondria have DNA. Now that alone doesn't exclude this hypothesis, because one could imagine that maybe DNA from the nucleus went from the nucleus into the mitochondria. And as an interesting side note, we do know this actually occurs. This even occurs to this day. Our DNA, to a very limited extent, can move between the nucleus and the mitochondria, and from the mitochondria and the nucleus. But don't worry, it's not like happening rapidly all the time. But it is something that can happen. So the question is this how the mitochondria formed? Did it form from these membranes of the plasma membrane, and then independently evolve electron transport on the inner membrane, and then finally the DNA from the nucleus would have had to transfer over to the mitochondria? Like I said, some of the DNA, not all of the DNA. So when these 2 hypotheses were developed, the endosymbiotic hypothesis and the within eukaryote hypothesis, we didn't have the technology, the ability to do whole genome sequence analyses or to build phylogenetic trees in the way that we can today. And so what they did was they built a phylogenetic tree. And in this tree, they compared mitochondrial DNA to alpha proteobacterial DNA and to nuclear DNA of the eukaryote host. So the idea here is then, if the mitochondrial DNA has a closer relationship with the nuclear DNA than to the proteobacterial DNA, then that would help support the within eukaryote hypothesis. However, if the mitochondrial DNA showed a closer relationship with that of the alpha proteobacterial DNA than to the eukaryotic host DNA, then that would support the endosymbiotic hypothesis. Well, if you're keeping score at home, you probably could take a pretty good guess as to what the data showed, because I've kind of already showed my hand by saying that the endosymbiotic hypothesis is not a hypothesis. It's a theory, meaning that it is well established and well accepted within the scientific community. So what these data showed was that indeed mitochondrial DNA is more closely related to alpha proteobacterial DNA. Than to nuclear DNA. This final piece of evidence, along with the 6 other points I pointed out to you, plus some additional data that I didn't show you, pretty much showed that the within eukaryote hypothesis couldn't be correct. And as I said before, this and all the other data allowed the endosymbiotic hypothesis to rise to the level of an endosymbiotic theory to help explain how mitochondria evolved inside the eukaryotic cell. So in establishing the endosymbiotic theory to explain the evolution of mitochondria, led some to ask the question, could the endosymbiotic theory explain the evolution of chloroplasts? So this idea would claim that we have a eukaryotic cell here with a nucleus, and it would have already have engulfed the aerobic bacteria to become the mitochondria, because we have evidence that the mitochondria were formed before the chloroplast. So again, this is the mitochondria, and up here is the nucleus. And we'll talk about the nucleus in just a moment. So how did it get the chloroplast? That is, how did this cell become photosynthetic? Well, using this endosymbiotic theory to explain it, one could imagine how our good friend from our last unit, the famous cyanobacteria, might be involved, because cyanobacteria has photosynthetic ability. So it's not difficult to imagine that if a primitive eukaryotic cell engulfed a bacterium that would one day become the mitochondria, it's not that hard to imagine that it might also have engulfed a cyanobacteria that would one day become a chloroplast. And let's show that over here. So again, we still have our mitochondria here, and we still have our nucleus here, but now, due to this engulfing of this cyanobacteria, we now have a structure that will one day become a chloroplast. So is there evidence for this? Well, let's talk about some of the evidence then. All right, so the first line of evidence is that chloroplasts have the same bacterial-like characteristics seen in mitochondria. So those things I listed for the mitochondria are also true in chloroplasts. Chloroplasts are about the same size as a bacterium. Chloroplasts have ribosomes. Chloroplasts have their own DNA, their own genomes, that is, that are more similar to bacteria than to nuclear plant DNA. They have double membranes, and in some cases they have more than two membranes, but it's consistent with an endosymbiotic relationship. Now I want to list some other characteristics that support chloroplasts evolving through endosymbiosis. And I already sort of mentioned this, but the circular DNA found in chloroplasts is very similar to cyanobacteria DNA. Another piece of evidence is that some protists have chloroplast membranes with the same peptidoglycan components that we see in cyanobacteria. Similarly, as I said with the mitochondria, some cyanobacteria live in protists and animals today. So now, with a firm understanding of the endosymbiotic theory and how it explains the evolution of mitochondria and chloroplasts, scientists were curious as to when chloroplasts first formed and in which group. Because if we look over here at this phylogenetic tree, we can see that several of these lineages are photosynthetic. For instance, the glaucophyte algae, red algae, green algae, and land plants all have the ability to undergo photosynthesis, as do these other lineages, shown here with these blue arrows. Now, through various studies, we're pretty sure that the primary endosymbiotic event that occurred with the cyanobacteria occurred here, leading to this group right here, including the glaucophyte algae, the red algae, the green algae, and the land plants. So if the primary endosymbiotic event occurred here, how did organisms like the euglenoids, or these down here, gain the ability to have photosynthesis? Well, a lot of different ways could happen. They could have independently have engulfed another cyanobacteria. But by comparing the genomes of the chloroplasts in the green algae and the red algae, and looking at the structures of the chloroplasts in all of these different groups, the best hypothesis is that there was a secondary endosymbiotic event, where this eukaryotic cell here, that, as we've drawn before, has a nucleus, but it also has the mitochondrion and the chloroplast. And so it was the result of this primary endosymbiotic event. And it then gets engulfed by a different protist that would have a nucleus and would have the mitochondria. But it was not able to undergo photosynthesis. But upon engulfing this green algae, say, It then acquired the chloroplast. What happened in the nucleus and the mitochondria? Well, it's likely they were degraded and just lost. This is our best guess as to how these other lineages of eukaryotes gained the ability to undergo photosynthesis through secondary endosymbiotic events. All right, the last thing I want to talk about with you today is the evolution of the nucleus. Unlike our previous discussion of the mitochondria and chloroplast evolution, the data around what we think is happening with the evolution of the nucleus is not as strong. So we won't refer to this as a theory. This is still at that hypothesis level. But essentially the idea is that we start with a cell that contains chromosomal DNA, but it doesn't have a membrane around it yet. And then we end up with this cell here that has DNA around it, that contains the chromosomal DNA with a membrane around it. So how did we get here? Well, a leading idea is that over time, parts of the plasma membrane started forming these infolds. And then eventually what happened was they began to surround that DNA. In a fairly elaborate structure that still maintained these infolds. And in doing so, it protected this DNA. And maybe a better word is to say isolated. Now, this infolding that came from the endoplasmic reticulum gave rise to two different structures. It did give rise to the nuclear envelope, which sometimes we just call the nuclear membrane, but it also gave rise these infoldings here outside of the nuclear envelope would become the endoplasmic reticulum. Now, what evidence do we have to support this? Even though it's not considered a theory, we still have evidence. We're just gaining more evidence. So some evidence includes observing bacteria doing this today. We still see bacterial species having this invagination of the plasma membrane moving towards the DNA. So it's reasonable to think that if we're seeing it today, that maybe it happened a billion or more years ago as well. And another observation is that the ER and nuclear envelope are still interconnected in our cells. So the last thing I want to talk about here is the advantage. Why do this? There has to be some sort of selective advantage that provided some increased fitness for these cells by creating this membrane around the DNA. One thing it does is it separates transcription and translation. So we haven't talked about transcription and translation in this class, and we really won't spend too much time talking about it. When you take cell biology and genetics, you'll spend a lot more time with it. But in bacteria, transcription and translation all occur within the cytoplasm. In eukaryotic cells, transcription, the making of RNA, occurs only in the nucleus, and translation, the making of the proteins, only occurs in the cytoplasm. This all leads to greater levels of gene regulation. Okay, that's all I have for today's video podcast. If you have any questions at all, please make sure you come see me or send me an email. If not, I will see you in class. Bye for now.